afternoon to everybody, and thank you for kind invitation. I'm really happy today to share with you my recently research interest on gene editing and gene editing strategy apply on epidermolysis bullosa. I would like to underline that it's not today I will show you some data and some results on a B simplex form that we reached in the last 2 years. And but I would like to underline that I'm working also on dystrophic EB due to genetic variation collagen 7 and also in another genodermatosis caused by genetic variation in a transcription factor TP63 that causes the EC syndrome. But today I want to discuss on simplex form and which is the next step for gene editing for epidermolysis bullosa in specifically AB simplex form. But where we are, my laboratory is placed in center of Italy, specifically in Modena. And I know that most in the world, this city is known for the Ferrari headquarters or the home of tortellini. But it's not in the scientific community. It's known also for another important achievement reached by the University of Modena and Reggio Emilia, and specifically by the spin-off in the Center for Regenerative Medicine Stefano Ferrari, the spin-off of the University of Modena and Reggio Emilia named Holostem, that reached a really important point, a really important achievement, because registered the first advanced medicinal product entirely based by autologous stem cell. The name is Holoclar. This important achievement is reached 11 years ago, thanks to the collaboration of University of Modena Reggio Emilia and the joint collaboration of the Chiesi Farmaceutici that more than 15 years ago building a new company. The name is Holostem. And thanks to the scientific support, and this is also the founder of the Holostem, thanks to Michele De Luca and Graziella Pellegrini, they established this technique in this center and obtain the first medicine, the approval of this type of treatment, this type of a product for the treatment of and the regeneration of corneal surface in a patients affected by ocular burns by physical or chemical injury. This is the scheme of product. It's autologous product, and it's useful for patients that receive a chemical or physical injury in monolateral A, from the contralateral A, healthy A, starting from a very small biopsy, less than 2 millimeters. We are able to collect the limbal stem cells and grow these cells in culture and generate a sheet of limbal culture. And we apply this sheet on the hay of patients affected by physical or chemical burns. This is the image of the sheet. And as you can see here, this is the image of the patients affected, the hay of patients affected before transplantation. They lose the stem cell, limbal stem cells, and for this reason, they have no regeneration in the cornea. So the central cornea was not transparent due to the growth of limbal stem cells. And the conjunctiva take over on the central cornea and cause this phenotype. The patients refer severe symptoms and the loss of visual acuity. And using Holoclar, this is the name of this type of product, autologous product, that we are able to remove the conjunctival pannus in these patients and apply the epithelium, the new corneal surface regenerating culture on this hay. And as you can see here, we have a full restoration of the visual acuity in this patient. This is only a few image of the patients that are treated with Holoclan, and a lot of patients are treated in Europe and UK territories, and now is commercialized by Holostem and distributed across Europe and UK territories. So this is an important achievement reached by Holostem and the University of Modena Reggio Emilia, but it's not the only achievement reached by this center. It's another important story was the story on regeneration of the skin in patients affected by junctional epidermolysis bullosa. This story started from a study started a lot of years ago from Michele De Luca, my mentor, at the beginning of 2000 years, when we treated for the first time a patient affected by junctional epidermolysis bullosa using a combined approach, a cell and gene therapy approach, and restore the skin in a patient affected by junctional epidermolysis bullosa. After the first patient treated, Michele and his team, and I have the opportunity to take part to this story during my postdoc. We treated additional two patients. This is the image of the last patient treated in 2015. The idea started, it's the same of Holoclar. We started from, it's an autologous product. It's an ex vivo gene therapy approach, starting from a small biopsy directly from the AB patient. Starting from this biopsy, the dimension of this biopsy is less than 2 centimeters square. And from this biopsy, we are able to collect epidermal cells that contain stem cells, and we transduce these cells with a retroviral vector. Why we decide to use the retroviral vector? Because it's an autosomal recessive disorder, and using a retroviral vector, we introduce in the cells a healthy copy of the gene, and in this way, we restore the expression of the mutant gene and the protein, and rescue the phenotype in the cells. After transduction, we generate epidermal graft and apply this graft directly on the patient. This is the image of the entire procedure. This is the story, the image of the patient at the beginning. He is a 7 years. He was a 7 years boy because now it's grown. He grew, and during at the beginning, these patients have most of the skin lose the epidermis, and 80% of his body is completely lack the epidermal sheet, the epidermis. So after stabilization of the patients, we transplanted this patient in a three different surgical procedure and applied the graft in the arms, in the leg, and in the back skin during three different surgical procedures. This is the image of patients after 1 year of follow-up, and as you can see here, there are no blister formation, no wounds, no any evidence of junctional epidermolysis bullosa. And we have a deposition, we observe a deposition of laminin 332 beta 3 that is affected in this patient and restore the adhesion property in this patient. But what it's really important for us and to understand that and formally demonstrate that we obtain a fully recover and a long-term follow-up because we transduce the stem cells at the beginning of this procedure. Because after 5 years of follow-up, we took a small biopsy and analyzed the expression of transgene expression in these cells. And as you can see here, we observe the expression of transgene in the whole biopsy that we took from patients after 5 years of follow-up. And this expression of the beta-3 chain of laminin 5 that we observe after 5 years of follow-up derive from transgenic skin. So this is a proof and evidence, and it's a formal evidence that we maintain this the clinical effect because we transduce at the beginning the stem cell compartment, and we analyze this after initially demonstrating that the stem cells are transduced, and we are able to maintain these cells after engraftment. So this is an important question, especially for a clinical point of view, because If we try to develop any gene therapy approach for epidermolysis bullosa, we take in consideration that the skin is a highly renewal tissue. So it's really important to target the stem cell compartment to obtain a clinical efficacy for the entire life of this patient. I want to underline that in this patient, we perform three different surgical transplantation, but to cover the entire body of this patient, the 80% of the body of this patient. But after this surgical procedure, we did not apply any other treatment on this patient. We did not apply any other graft because the skin is completely normal. Indeed, after 5 years of follow-up, we observed that the no blister are observed in transplanted area. No need for multiple graft is needed in these patients. The patient refer normal wound healing upon injury. This is an important point because this patient, when received this treatment, had 7 years ago, 7 years old. But during the life, these patients have a normal life up to now, and during the play, usually take some injuries. And he referred no wound healing and no blister after injury. In addition, we observe also no adverse events in the skin because we use a retroviral vector and should be in principle could be not so safe. But we have evidence that the use of retroviral vector is safe for the skin and for transgen for a gene therapy approach. It's not only our evidence because also the Stanford group in US that received approval from of the first treatment for dystrophic EB based on the same approach that we developed for junctional EB demonstrate that it's safe. So there is no problem in the use of retroviral vector for gene therapy approach. Normal nociception was referred, no pain, no itch. And in addition, no need for ointment or cream in this patient. The skin is completely normal. This is the image of patients after 8 years of follow-up. We receive the update from patient every year. And as you can see, we treated the arm, the leg, and the back skin. And as you can see, this is the image of the beginning. This is the skin after 8 years of transplantation, completely normal. This is an ex vivo gene therapy approach for autosomal recessive form and take in consideration that this is a very severe phenotype. These patients have show severe phenotype and this feasible, this type of approach, especially for severe AB form. There is a taking, we need this is feasible, this approach is feasible, scalable. From my point of view, complete is yes. We obtain a permanent skin restoration by single treatment and we apply multiple administration during 1 year. This is it's a really important point because in principle, we can, for any patients affected by a junctional epidermolysis bullosa, we can obtain a progressive substitution of the epidermis during the life of patient based on need of patient. We use integrating viral vector, yes, but it's safe. We need, and this type of procedure, it's mandatory, the surgical procedure. It's cumbersome per patient, but it's, yes, probably it's a surgical procedure, of course, but we restore entire the skin in these patients. And it's finally, this is the main point that I consider that not be can address by this type of a procedure. This is cumbersome, especially for a specific body site, for example, the periorificial body site. So this is it's not only the ex vivo gene therapy approach developed for epidermolysis bullosa, because using the same approach that we did for junctional AB, the Stanford group, thanks to the collaboration with Abeona, developed and received the approval for commercialization, for the marketing authorization from another treatment that is the same that we did for junctional EB. That in this case, they used a retroviral vector carrying the coding sequence of collagen 7A1. This is the first cell-based gene therapy approach product for patients with recessive dystrophic epidermolysis bullosa approved the last years. Again, this approach demonstrated it's safe and it's feasible, especially for patients affected by severe form of epidermolysis bullosa and specifically for autosomal recessive form. This type of approach is feasible for dominant EB form, for dominant mutation? The answer is no, because it's impossible to restore the function of dominant mutation using using a retroviral vector. In other way, adding a healthy copy of the gene in the cells. We need to remove the mutant allele or silence the mutant allele to restore the function, to restore the expression of the protein and the function in the cells. So our idea now is start with a different approach, completely different approach compared to with the approach developed for junctional epidermolysis bullosa, and work on simplex form due to mutation in keratin 14. So patients affected by simplex form due to genetic variation keratin 14, the genetic variation it's only in one allele. The other one allele maintain the healthy copy of the gene. So in this case, the patients show genetic change. The genetic change leads to keratin intermediate filament disruption, as you can see here is a simplified version. Now our idea is to use a gene editing tool based on a CRISPR-Cas9 system to silence specifically the mutant allele and restore the expression of intermediate filament in these cells. In this way, we disrupt the coding sequence keratin 14 using the SPCas9 and restore and maintain the expression only of the wild type allele in these cells. So starting from this idea, Camila Cattaneo, 2 years ago, with the collaboration of all the team in the Lucas Lab, developed this type of approach and demonstrate that this approach is feasible for patients affected by keratin 14 genetic variation that cause epidermolysis bullosa simplex. We received a biopsy from a patient, AB simplex patients that carrying this type of altered genetic variation, a deletion of this region in the genomic sequence, and it's quite easy to design a guide RNA that is able to discriminate the mutant allele and that do not recognize the wild type 1. So in this way, using a SPCas9 guide RNA, we introduce a double strand break in the mutant allele and induce indels after the use of a CRISPR system. And in this way, we obtain alteration the coding on the coding reading frame of the keratin 14 mutant allele that cause the premature stop codon, and this allele is silenced. So we introduced the editing machinery using by electroporation. And as you can see here, on target editing demonstrate the high efficiency of editing only on the mutant allele, leave intact the wild type 1. If you consider in the editing after the different indels on the on target, we observe that most of them are deletion, the other one are insertion and substitution. And this is all of these cause a frame shift and a premature stop codon in the keratin 14 mutant allele. Regarding the keratin 14 expression, as you can see here, we observe that keratinocytes derived from a B patient, a B simplex patient, show an accumulation of the keratin 14 expression. This is due to the alteration in the protein. And after the editing, we observe a restoration of the expression keratin 14 in the cells. But what's happened to the intermediate filament? After the genetic, as you can see here, we observe in keratinocytes derived from simplex from ABS patient, dots of the intermediate filament, keratin 14 in adults. It's typical. This is usually phenotype of simple keratin 14 alteration. After the editing, as you can see here, we observe a fully restoration of intermediate filament. This is happening on the cells because we obtain more than 90% of editing efficiency. To elicit this type of effect and to understand if the mechanical restoration of intermediate filament occurs properly, we apply to the cells a heat shock. We introduce this type of assay, an heat shock assay, that induce the destruction of intermediate filament, and after time, after different time, we observe a restoration of the intermediate filament structure. And as you can see here, now in the simple keratinocyte derived from patient, we observe that the intermediate filaments cannot be restored, and in the edited cells, we observe a full restoration of intermediate filament. But another important point is to understand if we have mechanical integrity in the skin, and to understand if we have a mechanical integrity, we decide to start to perform the 3D full skin model, a 3D skin full reconstitution, full thickness skin reconstitution in vitro, and starting from keratinocytes derived from AB simplex patients, we reconstructed the skin, and as you can see here, we observe a lot of blister detachment of the epidermis from the dermis. But in the same model, using cells, edited cells, as you can see here, we observe no blister, no alteration in the adhesion property. So the restoration of mechanical integrity occurs after editing. In terms of off-target activity, because we use a CRISPR system, CRISPR-Cas9, to induce the knockout of the mutant, specific knockout of the mutant allele, we performed a different analysis to analyze the off-target effect of the guide on the genome. And as you can see here, by guide RNA-seq, we observed some off-target, but most of them are in intronic region. Only one gene, only one off-target is recovered in an exon of a specific gene ZNRF320. And confirm this data, but confirm this data not only by RNA-seq, but also by NGS analysis. This is the only off-target, but there is no effect on keratinocytes. Regarding the last point, as I told you before, it's really important to understand that we are able to target the stem cell compartment, because in a clinical perspective, it's very important. It's really important to understand if this type of procedure that take in consideration the use of electroporation on the cells did not alter the stem cell compartment. And for this reason, we decided to analyze in a different way by clonal analysis that specifically identify the stem cells, the holoclones in red, and transient amplified progenitors in blue and green. And this is the meroclone and paraclone are transient amplified progenitors. And as you can see, the before and after gene editing, we observe the same distribution in terms of stem cell and transient amplified progenitor, and the stem cells are not affected by electroporation. These data were confirmed also by single cell RNA-seq data. And based on we recovered the DNA from the holoclones, the stem cell compartment in primary keratinocytes. And as you can see here, we observe that all holoclones are edited, and there are no expression of the mutant allele. So this is information, it's really important for any consideration of this type of approach for clinical application. But this work is published on molecular therapy, and we are able to underline and demonstrate that it works. However, it represents a personalized gene editing strategy. And in principle, to go on with this type of procedure, we need to design a specific guide RNA, a specific approach for each mutation for each mutation. So this is a personalized approach and it's not so feasible. In any case, I think that all of you on in the scientific community, it's most famous this story that take place last year's, because this is it's the first in vivo gene editing approach approved in only 8 months. It's a very personalized approach. But this achievement is reached only because a lot of people, university, company, and the continuum collaboration with the FDA was take place, and it's the achievement that probably cannot be replicated in all rare disease. So, taking consideration this point, I would like also to underline that it's very hard to translate any cell on gene therapy approach specifically for rare disease from basic research to clinical application, specifically to give a chance to this type of approach to receive a marketing authorization and belongs as sort of a gene therapy approach for patients affected by epidermolysis bullosa. This is impressive because, as you can see here in blue, the line in blue describe the preclinical studies. It's a lot of, the number is too high. But if you consider the phase 1, 2, and 3, and especially pre-registration phase, the number is too low. The problem is due to the sustainability of this type of approach. So the identification of personalized approach that it's dedicated for each specific mutation, I think it should be not very easy to manage. And I want to underline that also in my experience, it's true, this story, because we demonstrated that we are able to treat patients affected by junctional epidermolysis bullosa using a genetically modified epidermal graft. We treated 3 patients. I have evidence that in all 3 patients, the skin are completely restored and it works at clinical level. But up to now, we do not reach the market access, and we do not reach any patients instead of the 3 patients treated up to now. So, taking consideration this information, and also another important information, especially for keratin and specifically for keratin 14 and keratin 5, the high sequence homology across different keratins take very difficult to design specific guide RNA for keratin 14 or mutation on keratin 5 mutation, because most of them recognize also other keratins specifically, and it's really difficult to identify a specific strategy that do not cause any off target on other keratins. So for this reason, I decided to start a new project, completely another approach based again on gene editing, but this approach relies on HDR-mediated gene editing for a simplex form. My idea is to substitute, to induce the double strand break in the keratin 14 locus, specifically in exon 1, and replace by donor template the entire coding sequence on keratin 14. This is the idea, because starting from just to see the mutation across the gene, because there are a lot of mutations across the gene, the keratin 14 gene, not only in one exon, but in the entire exon in the gene. So it's impossible to identify, to create a sort of a lot of different approach for each mutation. In this way, what we are doing, we introduce in a double strand break in intro 1, and thanks to a donor template delivered by a V vector, we induce the HDR-mediated gene editing and insert the coding sequence of the keratin 14, the entire coding sequence of keratin 14, under the endogenous promoter. In this way, with a single strategy, we are developing a single strategy for a multiple ABS patient. And this is feasible. Probably it's more easy to develop this type of approach also for a clinical point of view. So this is the idea and the scheme that Silvia Zacchino, the PhD student in my lab, started 1 year ago, and she tried to demonstrate. They design a new donor template that carry an EGFP fused with the exon 1 codon optimized. This is due to these strategies has been designed in order to verify very quickly if the knock-in strategy work or not in our cells. And this is our preliminary data, and we introduce the donor template by AV vector and induce the double-strand break by SPCAS9 plus guide RNA. And as you can see here, we obtain a high efficiency of gene editing the cells in ABS context. And as you can see here, we observe also the full restoration of intermediate filament in keratin in this context, in this background. So this story, it's at the beginning, and more and more will be due. And thanks to the support of Deborah O'Kay, we receive a non-clinical PhD studentship for the next year, from that starting this year, and we are seeking a PhD student. So regarding the approach, this is the one main point that I would like to underline with you. Up to now, we developed some ex vivo gene therapy approach, not only for junctional EB, but also for dystrophic EB due to genetic variation collagen 7. But the ex vivo approach, an ex vivo gene therapy approach, I think it should be feasible for severe form. It's less for a mild phenotype or for specifically, for example, for simplex form or in general for other AB form. So my idea is to change my mind to my approach to ex vivo gene therapy approach to in vivo gene therapy approach based on delivering of gene editing machinery by LNPs. This is what we are doing in the lab up to now, and we are working very hard to do this aim in my team. Why? Because it's less cumbersome for patients. Because it's the genetic approach. This approach relies on the use of genetic approach and not non-integrated viral vector. It's possible to have a multiple topical administration, and potentially it's a permanent effect, especially if we target the stem cell compartment. And outpatient administration will simplify logistics. So this is the reason that now guide me in my new research interest in the future and up to now and in the future. So before to finish, I have to thank all the people in these slides, and especially my mentor, Professor Michele De Luca, that he support me during my postdoc, also now in my research interest. We collaborate every day to foster the development of any cell gene therapy and gene therapy approach for epidermolysis bullosa. All the members in the Lucas lab, but I need to thank my group, Alessandra Fabriz, my postdoc that work on dystrophic EB and the EC syndrome, Silvia Zacchino that works on simplex form, and Katia Russo and Marta Valenti that work on simplex on dystrophic EB and EC syndrome. And all the collaborators mentioned in this slide, and Camila Cattaneo, former member that works on the paper published on molecular therapy. Thank you for your attention. I'm waiting for your question.
Thank you so much, Laura. It truly is a a ray of hope, I think, to see that this is possible. It's possible. It is possible. It's just difficult. Is that a fair thing
It's
to
difficult,
say?
but it's possible. It's difficult, but it's possible.
Okay. We have one question already asking how to deliver the gene editing box to keratinocytes, and was
My
there a high cell death rate if using electroporation? So, people, if we've got some patients or members on the call, electroporation is a way of getting the gene into cells, isn't it, using electricity?
Yeah,
Yeah,
exactly.
yeah. So
Yes.
it's a, yeah.
We use electroporation to introduce a very quick, short electricity step in the cells that induce the open of the pore in the membrane, and in this way we obtain the machinery, the RNP, the Cas9 machinery, the genetic machinery goes in the cells.
And does it kill a lot of the cells? I'm thinking this
No,
is no,
not so much. No.
but it's not something you do to people. This is something for the ex vivo when we give.
This is the very it's this is the new aim, and up to now there are a few evidence that it's possible to deliver base editing approach, base editing machinery on the skin. It's published recently from other research in the world, specifically from Sarah Hendrick, that demonstrate that it's possible to deliver base editing directly in vivo in the skin. So it's an open question. My main interest is to understand if we reach the stem cell compartment, because I think it's really important to reach the stem cell compartment also by ex vivo, by in vivo approach, because in this way, this is the real chance for patients, I think.
I think everyone has this dream that they could apply something to the skin and it would correct that gene. So um, I guess that's yeah, that's what everybody would like. When you talk about the stem cell compartment, am I right in thinking that's quite? Are you thinking that that's at the bottom of the epidermis, sort of at the basement membrane?
And the base membrane is the
Yes.
first layer of the epidermis, contain the stem cells. It's interspersed in the basal layer.
So methods for getting it in. Would there be an idea that if there was already a wound present, that would get you
Sorry, I
there?
didn't understand.
If there is already a wound, if the epidermis is already traumatized, damaged, does
Mhm.
that help you to get access to the stem cell compartment? Or
My idea,
we've heard
yes,
lots of
this is
things
my
about
idea.
this is
The
yeah,
wound is my target for in vivo gene editing, because
yeah.
I think during the wound healing process, the cells grow and the stem cells is active, and I think it is the time. This is the time to correct the stem cells.
So I have a sort of a question. I'm not, you might have covered this, but certainly when the transplants were done for the skin of the boy with junctional EB,
Mhm.
um, presumably you can't cover the whole area. Once you've got some corrected stem cells in place, do you know if they spread? So as the skin with the EB skin maybe is damaged, does the replacement skin cells, do they do any spreading out and filling in the gaps? Or is that wishful
They
thinking?
spread only the gap.
Okay.
But if they taking in contact with intact skin, stop it spreading.
So, okay, okay. So,
Just we need to engraft the epidermis. For example, we apply the graft,
mhm,
and if the graft in the marginal region is open, they grow. But when
okay,
we obtain taking uh, taking contact with the epidermis in the border, they stop to spray.
okay. So they just fill in where they're needed to some extent.
Yeah.
Okay. It does sound like a fantastic hope for this. So I guess I don't want to be too a negative thing, but for some of the EB symptoms inside the body, is
Yes.
there a way of, in any way, translating this idea to? I know I'm jumping ahead. Let's get the one stage
No, no,
working.
yes,
But
yes. This is a really important point. Inside epithelium, how we can reach the inside epithelium, or we reached, and especially for example, the esophagus
yeah,
tract. I know.
yeah.
I think it's too hard to declare that now it's possible with LNPs. But
Yes.
in principle, it's safe. We can reach also in epithelium, internal epithelium. But it's important that it's safe and target specifically the epithelium and do not target, for example, liver. Because usually when we deliver the LNPs in the circulum, we
Yeah.
directly target the liver, not the
So
other, because
uh,
it's a typical of LNPs. This is due to the intrinsic nature of LNPs.
LNPs, these are the lipid
Lipid nanoparticles.
nanoparticles, little oily blobs. This is
Yeah.
the, yeah,
Lipid nanoparticles. Yeah.
instead of a virus. Or
Yeah.
yes, I'm just been
They
scrambling.
have a sort of uptake from liver. So it's really important
Okay,
to obtain to modify the LNPs, or in any case to have to ameliorate the targeting. Up to now,
yes.
there is no evidence. So I think it's the future, I hope.
It is very complicated. We've had a lovely comment in the chat. Someone saying it's not a question, but they'd like to say thank you to you and your team for the work you've done and continue to do, and that they've enjoyed listening to your explanations. I've really enjoyed your talk. Again, I wish I had that little bit more knowledge to understand it all. But to me, it does feel very hopeful and like the first hardest bit has been done to show that these things are possible, and then it's a case of translating it so it reaches more people. Is that fair, fair. Well, has the first hardest bit been done, or is there still another hurdle
It's
to
too hard
climb?
to reply to this question because I am no, it's too hard
It's
to
hard.
translate this type of approach to a broad range of patients. Very difficult, but not for a scientific point of view. It's
Yes.
only a problem regarding the sustainability in terms of cost, because it's too high. The cost to manage this type of product is very high. And it's impossible, and it's a rare disease. It's a rare
Yes.
disease, so there is a sort of problem specifically on the sustainability of this type of procedure.